Hello and assalamu alaikum. My name is Adan Iqbal. I welcome all of you. In this video, we are going to talk about the physiological salt solution. Now, what is physiological salt solutions? For example, if you want to study the tissue, if you want to study the animal tissue or the human tissue or the muscle outside the animal or the human body, then you have to place these tissues or the muscle in a liquid medium as that would prevent the drying or the damage of the tissue or the muscle which you want to study. So the liquid medium in which you are placing your tissue or the muscle for the study purposes, that liquid medium is known as the physiological salt solution. So the liquid medium or the fluids in which we are placing the animal tissue or the muscle which you want to study outside the living or the human body, that liquid medium or fluids are being known as the physiological salt solution. And why we call these liquid as a physiological salt solution? Because these liquid medium or the fluids have the same physiology of the fluids which are inside the living body. So they have the same, these fluids have the same isotonicity with the fluids inside the animal or the human body, and they have the same pH of the fluids as of the animal or the human body. So for example, our blood has the pH of 7.3. So these physiological salt solution have the pH of 7.3 also. So they have the same physiology of the fluids which are inside the animal or the human body. So this is the reason we call it as a physiological solution. And why we are also calling it a salt solution, because these liquid mediums are being prepared by the salts dissolved in the distilled water, and the salts like the sodium chloride, calcium chloride, and many other dissolved in the distilled water, and you get the physiological salt solution. So these liquid mediums are being known as the physiological salt solution. The reason being, they resemble the same physiology of the fluid which are inside the living body, and they are being prepared by the salt dissolved in distilled water. So this is the reason we are called these liquid medium as the physiological salt solution. Now the question arises, why there is a need to use the physiological salt solution when you are studying the tissue or the muscle outside the living body. So the answer is, that as you are taking out the muscle or the tissue outside the living body, so you have to give the environment which resemble inside the living body. And for one purpose, you give the physiological salt solution. Now in the physiological salt solution, we have the salts, we have the cations, we have the anions, we have the glucose. So these things will try to maintain the tissue in the viable state or in the living state. So if you do not use the physiological salt solution when you have to study the animal tissue, the muscle, your tissue will die off, your tissue will be damaged, your tissue will go into the necrosis. So you won't be able to find out any effect on the tissue or the muscle which you want to study. So the physiological salt solution, you must use the physiological salt solution if you want to study the animal tissue or the muscle outside the living body. So this is the importance of the physiological salt solution. And the first physiological salt solution is known as the ringer solution, because that was being formulated by the British physiologist S. Ringer. So on his name we call it as a ringer solution, and that ringer solution is the mixture of the sodium chloride, calcium chloride, sodium bicarbonate, and potassium chloride solution. So in the nutshell, what is physiological salt solution? Physiological salt solution is a salt solution in which you place your particular tissue or the muscle you want to study outside the living body, and that salt solution have the same physiology inside of the living body and have the same pH and have the same isotonicity. So the physiological salt solution are also being known as the PSS, or we also call the physiological salt solution as a ringer solution. And these are the solution of the salt or the salts that is essentially isotonic with the tissue fluids or the blood. And these physiological salt solution are also being known as a saline. They are also called as a brine. saline solution, normal salt solution, physiological saline solution, or physiological salt solution. So these liquid mediums have many names. They can be called by any name, and any name corresponds to the physiological salt solution. So if any names comes, if you read any name among these, that would come in your mind that these are the solution in which you have placed your animal tissue, the muscle which you want to study outside the living body. So, physiological salt solution, or the PSS, is artificially prepared solution. These are the solution which you prepared in the lab. You simply take out the salts and you weigh these salts and dissolve the distilled water, and you get the physiological salt solution. So these are the artificially prepared solution which are used to maintain the tissues in the viable or the living state. And PSS can be used to keep the isolated tissue alive under the experimental condition. And PSS is very important to maintain the tissue outside the animal body and fulfills their internal environment of the ions and the cations and the nutrition. As you know that your internal organs are being getting the anions and the cations like the sodium or the potassium. These are very important for you, and so therefore you want to give these anion and cation outside the living body, and these anion and cations are being given by the physiological salt solution. So in the physiological salt solution, we have many different types of the salt solution. Among them is the frog ringer solution, mammalian ringer solution, thyroid solution, crabs, hen psyllid salt solution, ringer loci solution, and D jalon solution. And these salt solutions are being prepared by the constituents which are being listed on the left side of the table. So these are all the constituents which are being added in the distilled water, or the deionized water, or the double distilled water, and you will get the respective salt solution. So among the constituents, we have the sodium chloride, potassium chloride, magnesium sulfate, sodium monophosphate, potassium dihydrogen phosphate, glucose, sodium carbonate, sodium lactate, and calcium chloride. So these are all the constituents, and these quantities are being for the 1 liter of the salt solution. For in order to prepare the 1 liter of frog ringer solution, you have to add these quantities in order to make the 1 liter of frog ringer solution. And by changing the quantities of these constituents, you will get a different salt solution. So for example, in the case of the frog ringer solution, you can, for example, if you only take the one constituents among the many constituents which we are add in the distilled water. So in the case of the frog ringer solution, you are adding 6.5 gram of sodium chloride in order to make 1 liter of frog ringer solution. But in the case of the mammalian ringer solution, the same sodium chloride you are adding is 9.2 gram. So the sodium chloride quantity is being changed for the frog ringer and the mammalian ringer solution. So different quantities are being used to make the different salt solution, and these quantities are being best suitable with the physiology of the fluid inside the animal body for which you have taken out the animal tissue or the muscle. So by changing the constituents, by changing the quantity of the constituent, you will get a different salt solution. So these all are the quantities for the 1 liter of the salt solution. So I have told you that the main components of the PSS are the sodium, potassium, calcium, glucose, magnesium chloride, and you can see that in these constituents we are all getting these main components. We have the glucose, we have the sodium, we have the potassium. So these are all the main constituents which you add in the salt solution. So these are the constituents or the main components of the PSS which are being used to prepare the physiological salt solution. Now we will talk about the role of these constituents. What is the role of these constituents in the physiological salt solution? And you know that all these are the constituents of the physiological salt solution: sodium, potassium, calcium, magnesium, bicarbonate, sodium, hydrogen phosphate, glucose, and the water, which we have discussed in these constituents. So what is the role of these constituents, which will briefly I will tell you that you know that the sodium is a major extracellular cation in your body. So, and sodium is always responsible for the excitation for the cell. Sodium is responsible for giving the excitation to the cell or the tissues which you want to study outside the living body. So therefore you are adding the sodium in the physiological salt solution. And potassium, you know that the potassium is a major intracellular cation inside the human body, and potassium is responsible for the nerve conduction and the muscle contraction. Calcium, you know that the calcium is responsible for the contraction of the muscle, so therefore you are adding the calcium. And you know that the magnesium is responsible for the relaxation. So the calcium and magnesium are always in reverse, or they have the opposite action to each other. Calcium is responsible for the contraction of the muscle, and magnesium is responsible for the relaxation of the muscle. And the bicarbonate and the sodium hydrogen phosphate, they are all being added to maintain the buffer, or they are added to maintain the pH of the physiological salt solution. And the glucose and water, they are being given to maintain the nutrition and the vehicle. So you can see that these are all the constituents of the physiological salt solution. And you know that these constituents are being inside the human body or the animal body, and these constituents are also being added in the physiological salt solution. So as the tissue or the muscle which you want to study outside the living body will get the same environment as the tissue or the muscle which are getting inside the animal or the human body. And these environment or the nutrition are being supplied by these important constituents which are also being inside the animal or the human body. So in making the physiological salt solution, there are some precautions which you have to take. You know that these are the constituents which you will add in the distilled water to make the physiological salt solution. So keeping in mind these constituents, the calcium chloride should be added in last in the physiological salt solution, and it should be added in small amounts. The reason being, calcium chloride tends to make the precipitates, and also it makes the solution turbid. So it's therefore better to add the calcium chloride in the last. Also, you know that the calcium chloride and magnesium chloride are hygroscopic in nature. It means they tend to absorb the moisture from the atmosphere. So it's better to make the calcium chloride and magnesium chloride stock solution separately, and then add the desired amount in the solution from these stock solutions. And if you have both magnesium chloride and magnesium sulfate, then it's better to add the magnesium sulfate, because magnesium sulfate tends to interfere less with the tissue activity, whereas the magnesium chloride tends to interfere with the tissue activity. So it's better to replace the magnesium chloride with the magnesium sulfate, because magnesium sulfate do not interfere, it interfere less with the tissue activity. So always prepare the fresh solution. The physiological salt solution should not be older than 24 hours, and it should be stored in the fridge in the temperature of 2 to 8 degrees centigrade. And the reason being, you do not use the physiological salt solution which is older than 24 hours, because there is a chance of the bacterial contamination in the salt solution, because in the solution you are adding the glucose. So the glucose is a nutrition for the bacteria, so it's better not to use the salt solution which is made older than 24 hours. Another term is the tissue aeration. Tissue aeration is the process of giving the oxygen and the carbon dioxide to the tissue or the muscles. And you know that the oxygen is being required by your cells, and without oxygen the cells will die. So your tissues or the muscles outside the living body will also need the oxygen, and that oxygen is being given to the muscle or the tissues, and along with that oxygen you are also giving the carbon dioxide. Now what is the reason of giving the carbon dioxide with the oxygen? You know that the tissue which you are studying, that tissue is being placed in the physiological salt solution, which is also known as the PSS. So the oxygen, if you give the oxygen will react with the bicarbonate which is present in the PSS, and that reaction of the oxygen with the bicarbonate in the PSS will result in the liberation or the removal of the carbon dioxide from the PSS. So when there will be a removal of the carbon dioxide from any solution, that will increase the pH of that particular solution. And when there is an increase of the pH, then there will be the alkalinity. So that removal of the carbon dioxide from the PSS will cause the solution alkaline. And so you do not have to change the pH, that will change the pH of the solution, and you don't want to change the pH of the solution. So what is the compensation? give the carbon dioxide from the outside, so that the removal of the carbon dioxide will be compensated from the carbon dioxide which you are giving from the outside. So that removal of the carbon dioxide will be overcome by giving the carbon dioxide from the outside. So this is the reason the carbon dioxide is given along with an oxygen, and the combination of the oxygen and the carbon dioxide is also known as the carbogen. So the carbogen is the combination of 95% oxygen and the 5% carbon dioxide, and the carbogen is being given to the tissue, and that is known as the tissue aeration. So you know that we have many different types of the physiological salt solution: frog ringer solution, thyroid solution, Crab's Gelen, MC1, and ringer Loki solution. And these physiological salt solution, particular individual salt solution is ideal for the specific tissue which you want to study. So for example, for the amphibian tissues, frog ringer solution is the ideal one. For the heart muscle studies, ringer Loki physiological salt solution is the ideal one. Crab solution is also known as the universal solution, because the crab solution can be used for any tissue or the muscles which you want to study outside the living body. In the MC, even along with the sodium chloride, potassium chloride, and other constituents, sucrose is also being there. But please correct it, the sucrose in the MC1 is not being present instead of glucose, because sucrose and glucose both are present in the MC1. And all the other remaining the PSS, all the other remaining physiological salt solution, they do not contain the sucrose. Only the MC1 contains the sucrose. So therefore, it is the reason they have highlighted it. But sucrose is not present instead of glucose. Both glucose and sucrose are present in the MC1. It is a very old physiological salt solution. For the avian SK muscles, which are the muscles of the bird, and the innervated muscles, the crabs, the D-gelen and the MC1 can be used. And for the smooth muscle studies, the thyroid solution can be used. So these salt solutions, they are being reserved for the particular tissues, uh, because these salt solutions are being best suited according to the particular tissue or the muscle which you try to study. So for example, the frog ringer solution, it is being reserved or ideal for the amphibian tissue studies. The reason being, the frog ringer solution is being best suited with the frog's body fluids. So for example, if you take out the tissue from the frog, and that particular tissue or the muscle outside the frog body will get the same body fluid environment in the form of the frog ringer solution, because in the frog ringer solution, the constituent which we add, they are being added with respect to the frog's body fluid composition. So these specific physiological salt solution, the constituent which we add, we have added these constituents according to the composition of the body fluids in that particular animal. So therefore, they are being reserved for the particular tissues.